DNA replication. The blue Sour Punch straws represent the DNA strands. Here, the topoisomerase, represented by the Twizzler, has unwinded the DNA strands and made visible the DNA bases. The red and orange M&Ms represent the same base. Next, helicase, represented by the candy corn, splits the two DNA strands apart. Then, single-strand binding proteins, as represented by the yellow M&Ms, attach to the separated DNA strands and prevent them from repairing. Then, primase, as represented by the Mike and Ike, will create a short piece of RNA primer, as represented by the Twizzler candy corn hybrid, onto the DNA for use as a template. The strand on top is the leading strand. Next, DNA polymerase III, as represented by the red gummy bear, will begin moving continuously down the leading strand, creating DNA. Onto the lagging strand. Primase will create an RNA primer onto the DNA strand. However, as DNA polymerase III begins to synthesize DNA, it will not be able to do so continuously. The fragments in DNA created are called Okazaki fragments. Next, DNA polymerase I, as represented by the green gummy bears, removes the RNA primers and creates a DNA strand in its place. Finally, DNA ligase, as represented by the jelly bean, connects the Okazaki fragments together as it passes down the lagging strand. At this time, DNA polymerase enzymes may also pass through to ensure for accuracy and proofread. From this, two daughter DNA strands are created. Well, that's it. I hope you enjoyed our little animation. The end.